So to finish up here with our neuromuscular junction, the junction between the neuron and the muscle fiber, and technically the end of the neuron, axon terminals, and what's called the motor end plate, which is the modified plasma membrane on the muscle fiber. What happens is acetylcholine, that neurotransmitter, is released by the axon terminals. That initiates a whole sequence of events within the muscle fiber that stimulates the muscle fiber, causing it to contract, and as we'll see, the entire muscle to contract. Blood vessels follow along with the nerves, and they supply oxygen and blood sugar glucose for the production of ATP by the numerous mitochondria, which we can see here, mitochondria, within the muscle fibers and with the muscle itself. Let's get into the microanatomy of the muscle fibers. The cells are really long and they're referred to as fibers. How long? Well, amazingly long for a cell. Most cells are microscopic. You would need a microscope to see them. And muscle fibers are microscopically thin, but they are very long. They run the length of the muscle, each individual muscle fiber, meaning muscle cell. So if you have a muscle that's, maybe a foot long, like your biceps brachii, there are muscle fibers that run the full length of the muscle. So that's very long for a muscle or individual cell, one reason why they're called muscle fibers. As we'll see when we look at the musculature of the leg, the longest muscle in your body is something called the sartorius, and it goes from your hip all the way down to your knee joint. So that's about 2 feet long, roughly. So very long, elongated muscle fibers. And muscles can have thousands of fibers depending on how big the muscle is, like a thigh muscle, with each fiber as long as the muscle. So the muscle fibers are formed from individual embryonic cells that fuse together. So they are multinucleate. As we see here, these little spots in purple, those are nuclei. So most cells in your body have one nucleus. Muscle fibers are unique in skeletal muscle where they're multinucleate. Let's see here. So what we see here, one muscle, and what sticks out here is one muscle fascicle, and then what's sticking out here is one elongated muscle fiber. That's what we see here, one muscle fiber. And then zooming in to one muscle fiber, the striations are obvious. And these little bumps here are the nuclei. They're located usually just underneath the sarcolemma, which is the special name for the muscle fiber plasma membrane. And then we'll look at individually, there's smaller components within each muscle fiber. So the sarcolemma is the cell plasma membrane of a muscle fiber. The sarcoplasm is the cytoplasm of a muscle fiber. And then there's invaginations of the sarcolemma that go into the muscle fiber, and those elaborate invaginations here in yellow are called the T-tubules or transverse tubules. So you see there's like these little pits here. So the sarcolemma kind of dives in and forms these elaborate transverse tubules or T-tubules that run throughout the internal structure of a muscle fiber. So again, that's one muscle fiber, and zooming in close to within a muscle fiber is what we see here. So there are myofibrils, and then there are myofilaments. So this one thing that's sticking out here is a myofibril. So each muscle fiber is composed of many myofibrils within them, and each myofibril has smaller yet myofilaments, and these are the thick and thin filaments, or thick and thin myofilaments. And the myofibrils run the length of a muscle fiber. And again, a muscle fiber runs the length of a muscle. The convoluted endoplasmic reticulum. The smooth ER of a muscle fiber is called the sarcoplasmic reticulum. And that's what we see here in light blue. It's very elaborate. And it stores calcium ions. And where the sarcoplasmic reticulum butts up against the T-tubules, there's pouches or pockets of the sarcoplasmic reticulum, and those are referred to as the terminal cisternae. Should be labeled here. I guess not, but what is labeled here is what's called a triad. So what a triad is within a muscle fiber is triad meaning 3. One terminal cisterna here of the sarcoplasmic reticulum butting up against a T-terminal, and another terminal cisterna of the sarcoplasmic reticulum. So this arrangement of these 3 components is referred to as a triad. It's another view of one muscle fiber here. Notice one muscle fiber, part of a muscle fascicle, which is part of the entire muscle. So the myofilaments are the filaments within each myofibril. And there are multiple myofibrils within each muscle fiber. So initially this can get confusing. Maybe not even initially, but a good thing to do is get a blank piece of paper and draw out the anatomy of all these components of a muscle to learn these components. Okay, so the thin filaments, which aren't labeled here, but they would probably be the little red ones here. The thin filaments are made up of protein actin for the most part, and the thick filaments are made up mostly of myosin protein. So let's look at what's referred to as the sarcomere organization within a muscle fiber, but also within a myofibril. So the myofibrils have what are called sarcomeres. So these are units within the myofibrils within muscle fiber. So a sarcomere is the structural unit between one Z line to the next Z line. So if we zoom in to one myofibril here, magnify it, we can see in blue in this diagram the thick filaments, and in red the thin filaments. And what we see here is that the thin filaments run along and they are attached to what's called a Z line. So there's a Z line here, thin filaments coming out of each side, and in the picture here the thin filaments are overlapping the thick filaments in purple or blue, the thick filaments, and they are attached to what's called the M line. You can think of that as, say, the middle line. And then running along this next side, well, there's thin filaments again attaching to that Z line, and then that continues on to the other, what's called sarcomere, and these run the length of a myofilament. So the sarcomere is from Z line to Z line. So from here to there is one sarcomere. Okay? And this is the functional unit of muscle, of a muscle fiber or a muscle fibril that does the contraction work or movement that allows the entire myofibril and the entire muscle fiber and the entire muscle to shorten or contract. And the mechanism of contraction is how much these thin and thick filaments overlap. Here there's a lot of overlap, zone of overlap. So this would be analogous, or an example or a picture or a diagram of a sarcomere essentially in full contraction. Now all the sarcomeres that are running through a myofibril will contract at the same time. So the whole myofibril will contract. Myofibril is part of a muscle fiber. The whole muscle will contract. The whole muscle fiber will contract, and that's part of a fascicle and then part of a muscle, so the entire muscle will contract in unison. But we're zooming into the single individual functional unit of muscle that does the contracting, and that is a sarcomere. So the thick and thin filaments give muscle its striated appearance, skeletal muscle. And that's because the thicker filaments on the slide appear darker and the thin filaments appear lighter. So there's the dark band and there's the light band, and then dark band there and dark band there. As you see here, the dark band, light band, dark band, etc. So those bands, the A band, notice that's the band that comprises all of the thick filaments, the entire thick filaments. So think A for A band and A for dark. Right? One way to remember what is in an A band. Now the I band, as you see here, is the region of only thin filaments, and it's lighter in color. So think I band and the I in light. So this is lighter in color here, the I band, because it only has thin filaments. This is darker in color, the A band, because it has thick filaments. Now it will also have some thin filaments depending on the amount of overlap, but that simply adds to the darkness of the A band. So the thin filaments overlap thick filaments in the A band. How much depends on the degree of contraction. So if we look at a slide here, those are the nuclei, so that's one muscle fiber, that's going to be another one. This is this banding we see, the striations we see, and you see an overall dark band, that's the A band, and then you see an overall lighter band, that's the I band. Now, if you look closely in the middle of the I band, there's a little dark thin line, that's the Z line. But in most microscope pictures or diagrams, they don't show you this amount of detail, or you can't see this amount of detail, you just see the dark and light, dark, light, dark, light. Okay? But from that thin Z line in the middle of the I band, where's my cursor? To the next Z line in the middle of that I band, that's one sarcomere. And then another one, and another one, another one. So the arrangement of the thin and thick filaments relative to each other give the banding, striation appearance of skeletal muscle. And the striations are indicative of the amount, of the region of thick filaments and the region of thin filaments. And as we'll see, as the muscle contracts, there's a visual difference in the I band relative to the A band. And we'll get there in a few minutes. So the H band, notice right in the very center of the A band, it's the H band's mid-region, and it only contains thick filaments. There's no overlap of thin filaments. And that will change in size depending, the width of the H band will change in size, again, depending on the amount of contraction, similar to the I band. And we'll get there. The M band, the M line, notice right down the middle of the H band, there are proteins here that bind and tether the thick filaments together, and that's just referred to as the M line. Think of it as the middle of the sarcomere, maybe. And then the Z line here and here bisects right down the middle of the I band, and that demarks, or that's the boundary from one sarcomere to the next. Okay? Z line to the next Z line is one sarcomere. So the thin filaments are anchored to, I have Z discs there, that's another name for Z line. So Z line is also called the Z disc sometimes, but the thin filaments are bound to that, to the Z line, Z disc. And there's a protein called elastic proteins, or something called elastin, but there are elastic proteins, think of like rubber bands, that help with overstretch and recoil, and that's what you see in green here. And that's what gives the muscle the elasticity, the muscle fibers the elasticity, and technically the myofibrils their elasticity. So there's a protein called titin that anchors the thick filaments to the Z